Hey, in the last video we spent a little bit of time talking about the four classes of organic molecules, specifically carbohydrates, lipids, nucleic acids, and proteins, but we only talked about them really at kind of an introductory level, and so in this video we're going to specifically dive into the two classes, carbohydrates and lipids, in order to kind of more fully understand exactly what the molecular makeup of these types of organic molecules are, and then how that molecular makeup makes a specific function that allows them to function the way they do in the body or in your body. Okay, so let's get started. If you recall from the last video, we talked specifically about monosaccharides being the monomer of carbohydrates. Remember, a monomer is a single subunit which are put together in repeated fashion in order to make a polymer, which in carbohydrates case is a polysaccharide. But monosaccharides in carbohydrate functioning or in carbohydrate building, there are three specific monosaccharides that the IB syllabus has elected for you to know, or has identified or deemed important for you to know, and those three monosaccharides being glucose, fructose, and galactose. Now, glucose, fructose, and galactose are going to be utilized by enzymes in order to build up dimers, specifically disaccharides and polysaccharides, as we build our more complex carbohydrates, specifically to store energy short term. Okay? If we look into the specifics of these three molecules, specifically glucose, fructose, and galactose, glucose is going to be a hexose ring. Okay? Hexose ring being a 6-carbon ring. You'll see that this is a carbon 1, 2, 3, 4, and 5. The sixth bond in this ring structure is going to be an oxygen, okay? And so there is 6 carbons, or there are 6 carbons, and that's how we get our chemical formula of C6H12O6. Notice that every single one of these, all three, glucose, fructose, and galactose, are all going to have the exact same C6H12O6 chemical formula. They all have 6 carbons, they all have 12 hydrogens, they all have 6 oxygens. But the way in which those carbons are structured indicates that there should be a different structure, and there is, and that different structure is going to give rise to a different function. And so glucose, being structured the way it is really good at producing energy in the form of ATP, adenosine triphosphate, during the cell respiration pathway. Okay, so glucose is the main energy source for the conversion of ADP to ATP during cell respiration, and is really going to be or become not only the main energy source for cell respiration, but is going to be utilized as one of the most important base units for many of the complex polysaccharides that we're going to talk about later in this video. There are a lot of them. We're going to focus on 3: starch, glycogen, and cellulose. But as the glucose is digested, absorbed, and transported, it's going to enter or be transported to the liver, and then is going to be pumped into the blood, or is going to be diffused into the blood in order for it to travel to every single one of the cells within all the tissues of your body, and is going to then be the energy source for that cellular respiration pathway, and is later, as we talk about it in the in later videos, going to require an insulin, which is a protein that is going to help moderate or regulate the blood sugar content. But glucose is going to be basically stripped from a lot of the complex carbohydrates you eat on a daily basis, is going to be pumped to the liver. It's going to be synthesized and broken down into the main component of glucose, and then that glucose is going to be pumped into the blood in order for cell respiration to take place. Fructose is a plant-based or a plant-derived monosaccharide. It's found in a lot of fruits. It's what gives fruits their sweet aromas and sweet tastes. It is 1.2 times sweeter than sucrose, and sucrose is a disaccharide. We're going to talk about it in the next slide. Sucrose is table sugar. Both sucrose and fructose are plant-derived, and so fructose and sucrose obviously have similar names, but they both have similar names because they're both derived from the same origin, being fruits. Both of them are very sweet. They are both hexose rings, or I should say both glucose and fructose are hexose rings. They both have the same chemical formula, and fructose is metabolized in the liver like glucose, but the difference is fructose does not require insulin, and so it is more easily broken down by the body. Glucose does require insulin, and so obviously when you think about the onset of diabetes, or you think about diabetes being a more of a disease, we don't often associate fructose with any diabetic discussions. We refer to blood glucose levels when we talk about diabetes, and it's only related to whether or not insulin is required in order to break it down. Fructose is not. And the third variety is galactose, and galactose is found in mammalian milk, and so it's one of the monosaccharides that is used to build lactose, which is the disaccharide found in milk, which you may have heard of. I mean, there's a lot of people that are lactose intolerant, and so if you're lactose intolerant, you lack the enzyme lactase that is going to be needed in order to break down lactose into monosaccharides. But galactose is found in milk, and again, it's a hexose ring. It's made of the same C6H12O6 chemical formula. It's 1/3 as sweet as sucrose, which is why when you drink milk, it's often not sweet. Okay, there's a big difference in sweetness between milk and a lot of fruit juices or a lot of fruits. And so 1/3 as sweet as sucrose is galactose, and there are not very many large stores of pure galactose in nature, and that's because galactose is often paired with glucose to form the disaccharide lactose, which I said before is found in milk. Okay, but when lactase breaks down lactose once you've drinking or consumed milk, glucose and galactose, which are the two monosaccharides that are bonded together to form lactose, are going to be stripped apart into their individual monosaccharides components, and you're going to have a single galactose monosaccharide and a single glucose for every disaccharide found in that sample of milk. Okay, if we focus on how two monosaccharides link together, it is a condensation reaction, and I have zoomed in and focused two of these kind of highlighted circles on two different hydroxide groups, two different OH groups, and if you notice, as we progress this animation, we are going to strip an H2O molecule, meaning we're going to pull a full OH from one of these carbons. Remember, this is a carbon, this is a carbon, and so both of these carbons are going to be holding on to a hydroxide each. We're going to remove a full OH from one carbon. We're going to remove an H from this OH group. And when you bond an OH and an H together, you produce a molecule of H2O. And because we are removing some of these atoms, the oxygen that lost its hydrogen has to form another bond. And so it forms the bond with the adjacent monosaccharide, in this case forming a glycosidic bond, which is really important. It is a type of covalent bond. It's very strong. It is going to be the bond that holds two monosaccharides together, forming a double ringed structure called a disaccharide. And when we bond the H and OH together, we're going to remove those two different components as water, and the water is going to leave. Okay, that is called a condensation reaction, and the condensation reaction, which is a type of anabolic reaction, is going to be utilized to link monomers, in this case monosaccharides, which are smaller subunits, together to form a disaccharide or a polysaccharide, both of which are larger molecules and produce water as a byproduct. This water is just released as a byproduct of this particular condensation reaction. Okay, so we've talked about the monosaccharides. We know that glucose, fructose, and galactose are the three disaccharides, and so are the three monosaccharides. But when we start linking those two or two of the three monosaccharides in different concentrations together, we are going to produce three different disaccharides. The disaccharides being sucrose, lactose, and maltose. Okay? Notice that each of these three disaccharides have a different two monosaccharide composition. Okay? And it's kind of hard to distinguish. They're all kind of a green color. This kind of lime green color is a glucose. You'll notice that a glucose and fructose together constitute a sucrose. a galactose and glucose together make a lactose, and two glucose molecules together produce a maltose. Okay, and so we're going to talk about specific characteristics or specific functions associated with these three disaccharides. Sucrose, remember, we said was table sugar. Anytime you think in your mind about sugar, you're probably thinking about the white crystalline powder that is found on everybody's kitchen counter or kitchen table in order to think about sugar. Okay, there are a lot of sugars in nature, and sucrose is one of them, one of many. But sucrose is a disaccharide that is composed of glucose and fructose with a condensation reaction. Remember, condensation reactions are the reactions that are producing that glycosidic bond that is holding two monosaccharides together. It doesn't matter what the monosaccharides are. It's the same reaction and the same removal of an H2O in every case. Okay, we're going to do a condensation reaction, we're going to remove an H2O, and we're going to link two monosaccharides, regardless of what the monosaccharides are, together by the oxygen and a glycosidic bond. Okay, it's going to form a disaccharide. But sucrose is known as table sugar. Okay, it's very sweet. It occurs widely in plants. It is a plant-derived disaccharide. In fact, you can find high concentrations of table sugar or sucrose in things like sugarcane and sugar beets. Okay? In fact, sugarcane is one of those crop plants that are actually harvested to produce sucrose. And sucrose, when ingested, is going to be broken down into the two smaller components, glucose and fructose. Okay? Again, when you break down the sucrose, you're going to split this bond in half, or you're going to basically add water at that point. You're going to give an H back to this oxygen, you're going to give an OH back to one of the carbons, and you're going to split the two monosaccharides together, or you're going to split them apart. And once the glucose is split from the fructose, the glucose is going to have to travel to the liver. It's going to have to be broken down or kind of digested or handled by insulin. The fructose, remember, is just going to be metabolized without the liver and without insulin. Lactose is the milk sugar, okay, found in mammalian milk. 2 to 8% of the milk by weight is lactose. It is a very non-sweet sugar and requires a specific lactase enzyme that is secreted by your intestinal villi and is utilized to break down lactose into the simple sugars for absorption. Remember, again, like we talked about when we were discussing galactose, galactose and glucose make up the disaccharide lactose. Okay, remember, galactose is not found in large concentrations because it typically is bound to a glucose, which produces lactose. Okay, only found in mammalian milk. This sugar is really structured to allow young developing mammals to not only develop but flourish and grow. Maltose is another plant-derived disaccharide, but it's specifically found in germinating seeds and is a very, very good, completely glucose-dense molecule that is utilized as a starch store for very quick direct energy as the young germinating seed becomes a young and then eventually full-blown plant. Okay? Maltase, just like lactase, is an enzyme that is present in yeast, and it is utilized to break down maltose into glucose. Okay? So why would a seed need to keep large amounts of maltose in their seed structures? The answer is a seed requires a lot of energy, specifically a lot of ATP, in order to produce the seedling from the actual seed or bean. And as maltose is broken down, notice that it is just basically pure glucose that has been linked together, and so there's no wasted energy. As soon as you split this apart, you can utilize the two different glucose molecules immediately to produce energy, to produce ATP. And when we start thinking about linking more monomers, specifically more glucose or more fructose or more galactose, to a growing chain of monosaccharides, you're going to get a very large complex molecule called polysaccharides. Again, just like the last video, poly means many, saccharides mean sugar, And so the polysaccharides are large, complex, multiple sugars that are utilized in a structural or functional manner. Structural means that it provides structure to the cell, and functional refers to it providing short-term energy for the cell. Okay? Now, we know that animal cells do not contain cell walls, and only plant cells contain cell walls. And so cellulose is a plant-based polysaccharide that is utilized in cell wall construction for structure. Okay, we know that the cell wall is a structure that is on the outside of a cell membrane and gives rigidity to a stem of a plant. Functional polysaccharides would be things like starch and glycogen. Starch is the plant functional polysaccharide, and glycogen is the animal functional polysaccharide. Okay, you can see over here that there are very similar looking repeated chains of single rings, but there are some substantial differences between them. The starch molecule, if you notice, is a single strand of individual glucose molecules, and every single one of these carbon groups is found on the exact same side. They are always on the top in this case. Cellulose is very different in that it is a single chain of glucose molecules, but you'll notice that the carbon groups have been alternating between the top and bottom. Okay, we're going to talk about that in a minute. And then glycogen is different as well in that it produces or facilitates side branching. And so it not only has a single chain or a single rope of glucose molecules like the other ones, but it allows for side chains, which is going to allow for a different function. Okay, and again, we're going to talk about all these individually. We just want to look at all three of them right now holistically to see kind of some similarities and differences. Okay, each one of these rings is a glycosidic bond, just like we talked about. They were all formed with a condensation reaction, and so water molecule is lost at every bond or at every linking of the next monosaccharide. But let's talk about starch specifically. If we talk about starch, which remember is the functional polysaccharide found in plants only, you'll notice that there is a bunch of glucose molecules that are linked via alpha 1,4 glycosidic bonds. Okay, and that's going to be different than the other ones, so keep that in mind. This molecule is hydrophilic, which means it would be soluble in water, because water, remember, is polar, and hydrophilic molecules are going to interact well with water. However, starch is a very large molecule, and because it is too large, it will not usually dissolve in water. Okay, so the fact that starch is hydrophilic doesn't readily mean it's going to dissolve in water. It's just too big. Okay, so if starch was cut down into smaller fragments, then it would dissolve in water. Okay, typically sugar dissolves well in water, and so if we take these large starch molecules and we start breaking them into individual sucrose molecules, for instance, it will more easily dissolve in water. Okay, starch is just too big to dissolve in water. It is a functional polysaccharide found in plants, like I said, and the function of this particular functional polysaccharide is for short-term energy storage. Okay, it is very easy and very quick to add or remove glucose molecules to this growing chain of monosaccharides. Okay, this is just where we store energy short-term. These starch molecules are going to be held in storage organs like potatoes. Okay, a potato is a very interesting part of a plant. The potato is actually designed to be a storage organ for a potato plant. You'll know that potatoes are grown underground, and so they aren't a photosynthetic body or a photosynthetic organ of the plant. They are just a storage organ. And so as the plant photosynthesizes above ground, it's going to be sending a lot of the starch molecules that it's making via photosynthesis down the plant down the stalk or down the stem and is storing them in a file cabinet, so to speak, which is the potato. And so as it produces more starch, it's going to produce a bigger and bigger potato. Amylose is a sugar. Amylopectin is a sugar. Both of these are different forms of starch. Okay, and so what does that mean? Different forms of starch have a different function. You'll notice that amylose is just a long, slender, singular chain of glucose molecules, and amylopectin produces side chains, both of which are storage molecules for plants, but you'll notice that the shape is different. Amylose contains or produces a helical shape. Amylopectin, because it is branched, forms a more three-dimensional globular shape. Okay, both of which can be several thousand glucose molecules large, but you'll notice that the amylopectin, because of the side branching, will actually become much larger. In fact, it can be up to 200,000 individual glucose molecules, whereas amylose is typically kind of bottomed out or topped out at 3,000. Okay, notice the glucose molecules are oriented in the same direction. We talked about that early on. All of these glucose molecules are oriented in the same direction, and they have these carbon groups on the top, and because they're all on the same direction, they're all traveling the same direction, they're all on the same side. It's actually not really shown in this diagram, but it's going to cause this chain to curve into a more curved chain, which you can see here. Because they are constantly curving, it produces kind of this corkscrew or curlicue structure, whereas the amylopectin isn't necessarily that case because of some side chains that break up that helical shape. Okay, now if we focus on cellulose, cellulose again is found in plants, so this starch is found in plants, cellulose is found in plants, and the main difference between starch and cellulose is the fact that starch is functional. So it is a functional polysaccharide. It's used to store energy short term. Cellulose is a structural polysaccharide. And so the fact that we orient these in opposite directions makes a more rigid molecule. Okay, rigid molecule. We orient these carbon groups opposite of each other so that it doesn't curve as the starch molecule did. It almost keeps them from curving, and that produces a very straight, rigid, very tensely sound, if that's a word, molecule. Okay? Glucose molecules produced by plants during photosynthesis and are producing a structural polysaccharide in cellulose that is going to reinforce their cell walls. These are long fibers, and they form a crisscross mesh due to hydrogen bonding. Okay, so if we have a cellulose, 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 I know it looks like a tic-tac-toe board, but these interactions right here would be hydrogen bonds that are holding these individual rigid cellulose fibers together, and because we can keep these very rigidly packed together, it's going to create a very, very structurally sound lattice structure that is going to produce a lot of rigidity to plant tissues. Okay, glucose molecules are linked via beta 1,4 glycosidic bonds. The fact that they are beta means that they are opposite of each other. So this one's going to be oriented up, so to speak, and this one's down, up, down. And so we have beta 1,4 glycosidic bonds. All of these are going to be linked via a condensation reaction, just like our other starch molecule, but they are producing a different bond type. They are still glycosidic bonds, but they are beta 1,4 instead of alpha 1,4. Cellulose is probably the most abundant organic compound on Earth, and that's just based on the sheer number of plants and the amount of plant tissues. Okay? And these are insoluble in water due to the large size again. So not only are starch too large, or the starch molecules are too large to be soluble in water, but cellulose is too large as well. Again, notice that this is a straight chain, and that's because the glucose molecules are oriented in an opposite direction, up, down, up, down, as opposed to all up, which gives a curved nature or produces a curved molecule in starch. And now we focus on glycogen. Glycogen, remember, is our only animal example of the 3 polysaccharides we talked about. Glycogen is a functional polysaccharide found in animals because, again, we don't have cell walls. We don't need the same forms or same types of structural polysaccharides. But these glucose molecules are linked via alpha 1,4-glycosidic bonds again. Notice that if these side chains weren't here, every single one of these glucose molecules are oriented in the same direction. It's just that this carbon that was here is now bound to an oxygen and another chain here. So even though these carbon side groups are not on top as these are, it still is oriented in the same direction. Okay, so it's an alpha 1,4-glycosidic bonds. They are producing branched kind of side chains, if you will, and that's so that we can pack more glucose molecules per square inch or per square micrometer. These are very densely packed and are very huge molecules. Okay, 30,000 base units can typically be made into a single glycogen molecule, and these molecules are produced by animals and are the main form of short-term energy storage in a lot of the tissues throughout the body. But most of the glycogen that is stored within a human body or within a dog or within any organism is not necessarily in the tissue cells, which there is some. Most of the highest concentration of glycogen is in the liver. Okay, the liver is not only an organ that filters the blood, so to speak, and removes toxins from the blood, it also is a energy store or a glycogen store for the glycogen that is produced by those condensation reactions. Okay, so that kind of concludes the carbohydrate portion. I think we're going to cut the video off and we're going to kind of keep that by itself so that it doesn't get too long. We'll focus on lipids in the next video. So bring your questions to class. See ya.